Good afternoon, everyone, and thank you for joining us for Webinar Wednesdays with PKD Foundation Chief Scientific Officer, Dr. David Barron. We're excited to be here. As usual, we're live tweeting throughout the event. Be sure to follow us at PKD Foundation and tweet questions using the hashtag Webinar Wednesdays. We'll add your questions to the queue to be answered at the end. During the presentation, you can also type your comments or questions into the chat panel on the right side of your screen. We'll have a Q&A at the end to answer as many questions as we have time for. Please note that the PKD Foundation does not offer medical advice, and the information shared this afternoon is not intended to be a substitute for consultations with your health professional. Care and treatment decisions related to your health must be made in consultation with your health professional team. We will be recording this webinar and will archive that recording on our website within the next few days. Today, David will be talking about total kidney volume, or TKV, and its importance in PKD. And with that, I will hand it over to David.
Hello. So today, I hope that you will learn what total kidney volume is, and I should add right at the beginning, by total I mean both kidneys, not one kidney at a time. And I'd also like to talk a bit on why individual variation is important, that all of us, whether patients with PKD or not, have different sized kidneys. It may only be by a little bit. If you're a very tall person, it may be a little bit more, but we'll talk about variation. That's important not only in total kidney volume, but in biomedical topics in general. And we'll also talk a bit about what biomarkers are, and these are very important for the ongoing clinical trials of new therapies for PKD and future clinical trials for PKD, many of which I am really looking forward to. And finally, as I said, we'll discuss why total kidney volume and some other biomarkers are particularly important for the development of these new therapeutics that are on the horizon. So what is volume? The first thing to say about it is it's not weight. Volume is independent of the density. So if a kidney were made of lead, for example, it would be very heavy, but it would have the same volume if it were the same size. So one way of measuring volume is to drop the object that you're interested in into, in this case, you're looking at a graduated cylinder. That's a piece of glassware that scientists use to measure amounts of fluid. And when you drop a solid, this is in a different language, obviously. Solid is not misspelled. It's just, I think, French, but I'm not positive. If you drop, for example, a solid, let's say a kidney, and you look at how much the water level rises that's in that graduated cylinder. And you can see that when that solid in purple is dropped, that the amount or the apparent height of the water goes up by about 5 ticks on the graduated cylinder. And when you subtract the volume of water in the graduated cylinder from the volume of water that's measured after you drop the object of interest into that graduated cylinder, that gives you an estimate of the volume. So this is somewhat simple when you have a sphere or a square or a very simple geometric object, but the kidney, for one thing, is not one of those simply shaped objects. And more importantly, and most obviously, it's in a living human being, so you can't exactly drop it into a large graduated cylinder or a bucket of water and see how the water level changes. So mathematicians have developed formulas for measuring volume without resorting to the simple experiment that I showed you in the previous slide. But again, those formulas are for maybe more complexly shaped objects, but still not as irregular as a human kidney. So one has to resort to different techniques, and those are imaging techniques that are used in living patients and subjects. And because each patient is unique, some people actually only have one kidney shaped like a horseshoe. That's somewhat rare, but obviously very different than having two roughly kidney bean, and that's where the name kidney bean comes from, shaped organisms in organs in your abdomen. So no formulas are really reliable based on geometric shapes. So those would be rough estimates. So we use imaging. The cheapest way to do this, inexpensive perhaps is the better word, is ultrasound. And that can give your nephrologist or physician a rough idea of how big your kidneys are, but it really isn't a precise measurement of their actual volume. Magnetic resonance imaging, or MRIs, is an expensive way to do this, but it has the advantage that it can be quantitated and three-dimensional information can be extracted from it. And this is basically the gold standard for measuring total kidney volume. That MRI is done of both kidneys, and formulas that are computer-based and the machinery give you an estimate of the total kidney volume. CAT scans are another means of measuring kidney volume, but they do use radiation. So MRI is, especially in research in PKD, really the gold standard. And there have been many papers published actually on TKV, because it is, if not the most, one of the most important biomarkers for the study of polycystic kidney disease. So even this month there was an article published on how to get the most precise estimate of kidney volume using MRI. And the technique can also be used to look at the measurement of not only the total kidney volume, but the measurement of what % of that total volume is occupied by cysts. And the cyst volume is really important because that, if you think about it, is one of the real measures of the severity of the disease in any particular individual. So the typical measurement for volume is milliliters. That's small m, capital L for liter. And as I just mentioned, this number really characterizes, particularly if you have a value at one time and then a value at some time in the future, of how your kidneys are growing. But even if you only have one measurement, it tells you whether your kidneys are relatively larger for your age or relatively smaller for your age than the average. And that gives the nephrologist some estimate of how advanced your disease is. But kidney growth is unique to PKD. In most other chronic kidney diseases, the kidneys may stay the same size or they may even atrophy. Atrophy meaning they sort of scar up and actually become a little smaller. So really, TKV is central to polycystic kidney disease and not really all that relevant to other chronic kidney diseases. And this is because for PKD, cysts are the disease. And cysts grow inexorably with time. We are, of course, looking for therapies that will stop that growth, but at this point, there are therapies that can slow the growth of cysts, but none so far that can actually stop the cyst growth or reverse it. But that's what we're looking for. And the growth of cysts are most easily followed by increases in total kidney volume. So of the studies that have been done of PKD, many use TKV as the critical measurement to look at the effect of control of blood pressure or the effect of a drug. And these studies have been done and reported recently. And studies that are ongoing that we have talked about in previous webinars, MRI measurement of total kidney volume is an important part of those studies. So each kidney is a slightly different shape and it's in a slightly different location. You can see how The kidneys are connected to the aorta in the dark red and to the vena cava in blue, and they're not quite at the same height on each side of your body. There's some asymmetry there. But for the average adult without PKD, if you clench your fist, you have a pretty good idea of how big your kidneys are. However, when you look at this with PKD, and I'm always struck by these photographs, that when you look on the left side, you see that fist-like pale structure, that's a normal kidney, and you see what a polycystic kidney looks like. As a frame of reference, on the right side, you can see that polycystic kidneys can be as large as a football, and you have a pair of those. So you can imagine the space that that takes up in your abdomen. This is a famous diagram now. I've modified it a little bit. This is adapted from Dr. Jared Grantham, one of the co-founders of the PKD Foundation, together with Vicente Torres at the Mayo Clinic. And it shows, very importantly, that kidneys are growing from a young age in PKD, maybe from birth, maybe even in utero, but it's only at the age typically of 50 or 60 when you see on the red line, which is GFR, or glomerular filtration rate, which is a measure of how well your kidneys function, that you stay pretty normal in terms of this basic measurement of renal function often into your 50s, and I know some people, including in my family, particularly the women for some reason, who may live until their 70s before they go into renal failure. This despite the fact that your kidneys are growing with time. So for a long time, even though your kidneys are getting increasingly cystic and total kidney volume is rising and rising, because you're born with an excess of nephrons, and because individual nephrons that aren't affected by the cysts can work harder, you maintain normal function despite the fact that from a physical perspective or a histological perspective, your kidneys are getting increasingly damaged. And this is one of the conundrums of how to treat for PKD. Do you treat when GFR is declining, or do you treat while your kidney function is still normal? And this is one of the key questions facing research in PKD today, and I'm confident we're going to reach some common ground with the regulatory authorities like the FDA in the United States and the European Medicines Agency in the European Union on when to treat for the disease. Now we get to the question of variability. And this is really very important because I'm showing you two pictures of polycystic kidneys. I think the one on the right is probably a little fuzzy. It wasn't a high-resolution picture to begin with. But if you look at them, the one on the left is pretty uniformly cystic. The one on the right, particularly at the bottom, looks comparatively normal. And this is what is really why variability is so important, because although most people will have kidneys that look somewhat like the kidney on the left, there are a few individuals that have polycystic kidneys like on the right where a significant portion of the kidney seems to be unaffected by the cysts. And these may be patients who never do go into renal failure because of the asymmetric distribution of the cysts. But on average, polycystic kidneys look more like the one on the left than on the right. Nevertheless, variability is an immutable feature of life, and it is this type of variability that the regulatory agencies are sometimes concerned with. So Aristotle, the philosopher, liked to think of objects of having an inherent or ideal shape, that there is an ideal chair. You can see this in statues of human beings, in Roman or Greek culture. And Darwin really looked at it somewhat differently. When he was on the Galapagos Islands, for example, he noticed that all the finches had different sized beaks, and he began to realize that there isn't really an ideal shape for those finches. They're inherently variable, and for those of you wondering about the bolding and inherently, inherit is within that word. And of course, PKD is an inheritable disease, just as is beak size in those finches that Darwin so carefully documented when he was living in the Galapagos. And this leads to statistics, and I admit I glaze over when talking about statistics, but they're very important because, as I said, no two patients are alike. And I picked from a study of young Korean men, and this could be Japanese men, it could be North American men, it could be African men, it could be European women from a specific country. But in each case, if you measured the average length of their kidney, in centimeters in this case, or the volume of their kidneys, as probably determined by MRI, you get values that are typical of that population. And the important thing to look at is not that the average volume of the left kidney is 207.32 centimeters cubed. That's another term for milliliters. But the fact is that you have a plus-minus sign. And then you see another number, and that's the standard error of the total kidney volume. And by error, I don't mean that it's a mistake. It means that for any average population of Korean men, there will be some that have kidney volumes of 240 or 250, and others that have total kidney volume that's under that value. So it's called an error. That's a statistical term. There's also something called standard deviation, which is a larger number. And deviation does not mean deviant in the sense that most of you might think of it. It means that we all deviate to some extent from the mean. And that's the average value. So yes, there are probably one or two Korean men that have a left kidney that is exactly 207.32 centimeters cubed, but almost the rest of them have some value that's close to that, but not exactly that. And that's an important consideration because there are people that may have relatively small kidneys, but have relatively small bodies, and so their relative kidney volume, although small compared to the average, may be large for them. So all these things need to be taken into consideration. Day-to-day total kidney volume is not really important. What's really important is the rate of growth over time. So when you look at total kidney volume, in general, I think nephrologists would say if you have smaller kidneys, you have a better prognosis at that point in time. Slower rate of growth over time, if you have, for example, an MRI and then look at it a year later, if your kidneys have not grown much, that's a good prognosis. And generally, with smaller kidneys and slower growth, there will be generally fewer complications. And I should add that these complications can also be biomarkers like pain, bleeding, and infections. A worse prognosis is larger kidneys. So the total kidney volume is large based on your age. If you were to do, just as I described, an MRI at day 1 and then at day 365 and you saw a faster rate of growth, that's not a good prognosis. And there's probably associated with the faster growth and larger kidney an increased likelihood of these other aspects of the disease, such as pain, infection, and even cyst rupture. So I argued with Alexis for a while on why we have that water bottle over there.
Respectful banter.
And it says words and unwords, because let's face it, statistics, there's data, damn data, and lies. And you know, the statisticians sort of get hit with that joke. But what you're looking at is the average distribution of a value. So when you see that little u at the bottom, or a micron in Latin, that's the average. And on either side of the average are standard deviations. So in a given population, if it's normally distributed like this bell curve, this is called a bell curve, 68% of the population will be within one standard deviation of the mean. You'll capture 95% of the total population within 2 standard deviations of the mean, and 99.7% of everybody measured that are within 3 standard deviations of the mean. That 0.3% is still out there, and they can really complicate a study if that 0.3% is represented in one study group but not the other study group. For example, if the people who are 4 or 5 standard deviations different end up in the control group and not in the treated group, you can end up with a difficult to interpret study. So these kind of statistics, I won't bore you with them much more, are very important because they really in the end determine whether a study has successfully differentiated between the average in one group, namely a control group, and the average in the treated group, namely a new therapy for PKD. And if you look at data from the HALT trial, and I'm not going to try to take you through every aspect of this, but you see some curves, and you see a light blue curve that is the group treated with standard blood pressure lowering medications, and you see those T's. Those aren't actually T's. They mark the standard deviation of the mean, which is the little blue circle, and that's the average. So that means that of all the individuals, there's a range of values. And if you look at the dotted red line, that's with rigorous blood pressure control or low blood pressure, and you can see the red T's, they're sort of overlapping with the blue lines. But if you measure the statistical difference between those two lines, even though there are some patients with the standard blood pressure treatment that are no different than the people with the low blood pressure treatment, that statistically you can show that there is a difference by more rigorously treating blood pressure, and that total kidney volume, which is what is being measured here, actually is lower in those treated rigorously to have their blood pressure lowered. Now, this doesn't come without a penalty. There are some side effects with low blood pressure. Some of these people, if they stand up too quickly, might feel dizzy. Other people are fully accommodate the lower blood pressure. Matter of fact, most of them do. But so this is the example of if these two lines were much closer to one another, the statistical significance between the two lines might not be big enough for the FDA to accept these type of data. And it's really what you're looking at here that determines whether a clinical trial is going to be accepted by the regulatory authorities as showing a clinically significant difference. And when I use the word significant in this case, I mean both clinically and statistically, because when these trials are started, you state in advance what your expectation is, and if you don't meet that expectation, then the study is a failure, even if you show a trend for an effect. So in the development for new therapies for PKD, we want to use TKV, and by we, I guess I should say me, and hopefully other people out there that are interested in PKD, and that is because, as we showed you earlier with the Grantham and Torres graph, my personal opinion is that we want to treat when the kidneys are growing but kidney function is still normal. In other words, we want to treat and prevent growth of the kidneys while you still have normal kidney function so that you can preserve preserve that normal kidney function for as long as possible. But it is difficult from a regulatory perspective to make that argument because some patients will never go on to renal failure and others will, and it's difficult to predict who will and who won't. Although TKV, being a prognostic biomarker that's now approved by the FDA, that is the case, but the change and the size of the kidneys that they're looking for may mean that you're still treating a little too late in the disease. So we are hoping to make advances on total kidney volume as a prognostic biomarker, and hopefully someday, perhaps in combination with other biomarkers such as pain, infection, or hospitalizations, becomes a surrogate biomarker. In other words, if a drug shows a significant effect on TKV and total other biomarkers altogether, that even though kidney function has not changed much because it's basically normal early in the disease, that drug might gain approval, particularly if you could pick which patients are more likely than not to go on to end-stage renal failure. So when kidneys are already really quite large, much of the remaining kidney that is supporting normal kidney function is no longer available because it's fibrotic, damaged, or crushed between the cysts. And even with an effective drug, there's not enough kidney left to really, for that drug to have efficacious effect. So to try to summarize what I've said, I've repeated another slide of Taurus and Grantham, and I've shown three time intervals in which you might want to start treatment for polycystic kidney disease. So when is it most likely to succeed? I guess I've used a stoplight metaphor, and there's green, there's yellow, there's red, and you can see in the green it's probably very possible within 6 months to 2 years, which is when pivotal clinical trials, the length at which clinical trials are typically done for registration of a new drug, that you would see a significant difference in the rate of total kidney volume growth. However, GFR is pretty much a flat line, so it would be pretty difficult to see a change. Although if one did, that drug would certainly be approved. In the yellow, total kidney volume is still growing, as it always has been. However, GFR, a measure of kidney function, is beginning, I like to say, to fall off the cliff. It's no longer on the plateau. It's beginning to decrease. And this yellow box is actually where you're most likely to see an effect in a 6 month to 2 year pivotal clinical trial. It's also a time at which kidney damage due to the growing cysts is significant enough that you wonder whether the therapy, even though it does slow the growth of cysts, has been started in time to really prevent end-stage renal disease. And when you go into the red box, when you have fallen off the cliff and your estimated GFR, a measure of your kidney function, is really declining fairly rapidly now, and the kidneys are really big, it's unlikely that drugs are going to be effective at this point in the study. Now, again, I want to stress that this is my opinion. There are other scientists that share this opinion as well as physicians, but these are important regulatory and scientific questions as to when in the course of the disease the drugs can be used. Because remember, in the green box, that's my preference for a new therapy, but you would also be taking that drug for a long period of time, and all drugs, like all substances, have their pluses and minuses. In other words, you have to weigh the efficacy of the drug against the safety profile of the drug. So there are many arguments and regulations that favor orange, but I'm hoping that green is the new orange. So can green be the new surrogate for PKD prognosis? We are typically born with about 1 million nephrons, or about 500,000 per kidney. Just like variation that I mentioned, some people are born with 800,000 nephrons or less. Some people are born with 1.5 million nephrons. One can imagine that if you're lucky enough to have that kind of endowment, that your kidneys will be relatively resistant to disease because you have a lot of reserve. And we know that 1 million nephrons is more than enough, because for those that are living kidney donors, they obviously can do quite well on half the number of nephrons. So as land-dwelling animals, we have a lot of kidney reserve for those times when we don't have access to water. So life can be normal with 1 kidney. So renal reserve is pretty good. And for living donors out there, within a half hour by an as yet unknown mechanism, there's actually compensatory growth of the remaining kidney. And I think even in transplants of living kidneys or non-living donors, that those kidneys can grow as well. The other thing that kidneys can do when nephrons are damaged is hyperfiltration. In other words, the amount of blood flowing through individual nephrons to be filtered and cleaned can be increased by hormonal mechanisms, and that can keep your kidney function going. But like anything that's overworked, those nephrons eventually, to use a slang term, they suffer burnout, basically. So that means that considerable renal injury, and this isn't just for PKD, but it's for many renal diseases, can go on before there's a significant measurable decline in renal function. I was recently at a meeting of the National Kidney Foundation, and many of my PKD friends first learned of their PKD the day before they went on dialysis. So they literally did not know that they were in renal failure, and they find out. I was amazed by that story. That's and that's for diabetic nephropathy and other renal diseases as well. And that's because these people are not going to a nephrologist, and it may, values indicative of kidney function may not have been paid close attention to by the family doctor, or perhaps some people just had not gone to a doctor for quite a while. So therefore, intuitively, I think treatment before considerable renal injury has occurred is really what one wants, but as easy as that is to say, I've been in the drug industry long enough to know that chronic use drugs do have potential safety issues. So, total kidney volume is an early treatment biomarker. Biomarkers are of multiple types. TKV has been formally approved by the FDA as a prognostic biomarker. In other words, drug companies, if they want to test a new therapy for PKD, can use large kidney volume as a way to enrich their trial in patients that are likely to go on to renal failure. And by that, they're more likely to see a treatment difference between the control group and the treated group, because they're starting with kidneys that are already very large and likely very damaged. But there are a lot of presumptions around that. A more substantial biomarker would be a so-called surrogate biomarker. And this is like high blood pressure. You look at your pressure before and after a blood pressure treatment. That's a surrogate biomarker. That's telling the doctors looking at a new antihypertensive medication that somebody who has high blood pressure, after taking the drug in a double-blind study, it's shown that the treatment reduces that high blood pressure to normal blood pressure. But blood pressure is just a biomarker. It's indicative that that patient later in life, if not treated well, is likely to suffer cardiovascular diseases, but it's and that's prognostic, but it's also because of all the work that's been done around high blood pressure. This is a surrogate. It's also, as I mentioned, an enrichment tool for designing trials. But the key presumption here is that kidney damage in PKD is widespread before renal functional decline is evident. And I'm quoting from Dr. Grantham again, "It may be futile to administer such agents late in the course of ADPKD when a host of different processes have combined to produce the fibrotic end-stage kidney." And I think most physicians and scientists would agree that that end-stage kidney in polycystic kidney disease is really beyond a therapeutic treatment. So again, to stress Dr. Grantham's point, is that the cysts are the disease, and this gets back to measuring total kidney volume and how critical that is as a tool for exploring new therapies, many of which I hope will be entering clinical trials soon. So to make TKV stronger, since it's a structural biomarker but not a functional biomarker, in other words, it's not GFR, one can look at the effect of new therapies on high blood pressure or hypertension, the incidence of pain in your flanks or musculoskeletal pain from having those large kidneys near your spine and altering how your spinal mechanics are working, incidences of hematuria or blood in your urine, proteinuria, which is an important sign of problems with your kidney, urinary tract infections, which are more common in PKD even when kidney function is normal, cyst ruptures. I've had one. They're acutely painful, and they're very significant for PKD patients because if you need a transfusion, as I did, that makes a subsequent transplant a bit more difficult since you are sensitized to someone else's blood who provided that transfusion. So these all could be used to refine both TKV as a structural biomarker and GFR as a functional biomarker. So in conclusion, total kidney volume is currently a prognostic biomarker of polycystic kidney disease, but can it be more? Can it be a surrogate biomarker? And that's for future discussion among scientists and clinicians and regulators, and we hope that there will be progress on the use of TKV in this regard. One of the first laboratories that I led in academia a long time ago was called the Core Structure Function Laboratory, and that's exactly what this is all about. I was an electron microscopist and a light microscopist, and I looked at structure, and what I wanted to do and what I provided as a service for the pharmacologists that I work with was how is function correlated with structure. And I think this is really what it's all about. TKV is a really sophisticated and precise measurement of structure and what's going on in a polycystic kidney. GFR is a commonly accepted measure of function, although it has its issues. So these lead to the regulatory issues for chronic diseases like PKD. What are the endpoints in pivotal clinical trials? Can they be TKV alone, or do they have to be associated with a functional change? Obviously, long-term safety is important, but that's important for all drugs, whatever their indication. And are the effects sustainable? Perhaps a drug can be taken for 3 to 5 years, retard the growth of cysts, and that retardation of growth is maintained even after the drug is stopped. And that would be really ideal in that you're not risking the safety issues of any drug, but you have an effect that's sustained and delays the onset of end-stage renal disease. And perhaps the biomarker for TKV will be combined with other variables, and this is called multivariate, and that there will be a multivariate biomarker. These are sometimes difficult in that the gold standard pivotal clinical trial that is used to register a new drug is usually looking at one primary endpoint, and this would be a combination of endpoints. And finally, I should say that you can obviously tell that I have my particular opinions about treating earlier in the course of PKD rather than later, but I would remind everyone that not all PKD patients, particularly those that have mutations in PKD2 gene, not the PKD1 gene, which is about 85% of us, may never go into end-stage renal disease, and some people with PKD1 may never go into end-stage renal disease. So you could theoretically be treating somebody with a drug who might never go into renal failure even without the drug. And that's obviously not known for patients in their 20s. But I would maintain that a drug that has an excellent safety profile, in other words, its efficacy far outweighs its safety issues, that this issue of not being able to predict the future, which if you think about it is the case with many drugs, a lot of people who smoke will not get lung cancer. People with high blood pressure may never have a heart attack. It's hard to predict the future. So I don't think that this question can't be overcome. I think it can. So with that, I'd be happy to answer any questions and hope that you have some notion of total kidney volume, what it means for PKD patients, what variability is all about and why that matters. We're all our own unique selves, and with that, I'd be happy to answer any questions.
And questions they have. So you all have kept me very busy kind of keeping track of this. Thank you so much for your questions and for your attention. The overwhelming question we're getting from very early in the webinar through to the end is, should people be demanding a measure of their TKV from their nephrologist? And I would like to just say that David not only is our CSO, but he's also a PKD patient, and so he can answer some of these questions from a scientific background, but also just from a patient-to-patient perspective. So what do you think, David?
So I don't think I had an MRI until I was in my late 40s, actually. And my internist, we sort of learned together about PKD. I was his first patient with PKD. I think he's only had 2 now. We've sort of grown old together.
He was a nephrologist?
No, he was an internist.
Okay.
And he used to get an idea of my kidney size by being hands-on, and he could feel my kidneys beneath my rib cage. In normal individuals, that's not the case. And he did order an ultrasound, but this is really a discussion to have with your nephrologist. If they feel that their treatment, of your disease is best served by having a baseline MRI, then of course I would follow their advice, but I certainly can't give my own advice about that.
So if you, and I think this is a question for you, the patient, not you, the scientist, if you as a patient feel like it is important to have that measure in your file, and you have a nephrologist who is not necessarily as committed to that or as interested in ordering those tests, do you have just advice person to person? Is it something that you should push very hard for? Is it something that maybe could be useful in the future, or should you just kind of let it go and follow the advice of your nephrologist?
Well, from a selfish perspective, I think that there are studies even ongoing where total kidney volume is measured, and I think scientifically, if not from a particular patient perspective, it's important to have those measurements. MRI, however, continues to be expensive. I think that insurance would cover that for PKD patients. I'm not sure.
It's one of the questions we got.
But ultrasound, as I mentioned early in the talk, is a much cheaper way We talked about standard deviations and standard errors. They're higher for ultrasound, but ultrasound still gives you good information, and that would be a much less expensive baseline that gives you imaging not only of kidney size but of the cysts as well. So there are multiple approaches that a nephrologist might take in terms of how they choose to image your kidneys and when.
So we have someone who has a daughter who's 18 now, who has PKD, AD-PKD, and she's been diagnosed for quite a while. You know, the question, it's a similar question. Is it worthwhile to push for these tests so that a baseline can be established early on and then continue every so often throughout, especially with someone as young as 18? It might be helpful as we move toward a hopeful therapy in the coming years. I don't know. What do you think?
So it's interesting. I just had a call unrelated to this webinar of somebody interested in the use of the statin drugs, which are used to lower cholesterol. And it so happens that there was a study done in children and young adults age 8 to 22, and that did seem to show a modest effect. So if imaging and statin use were a therapeutic option, you know, for the nephrologist of that patient, of course. Establishing the baseline with these techniques, as I mentioned, can be expensive, but they're non-invasive, and there's really no risk attached to them, so it becomes a matter of, you know, the nephrologist's opinion and the cost.
So I think, and we have more questions, we'll get to them, but I just will say that this is, it sounds to me like something that as the foundation we need to look at holistically and maybe talk with some of our key physicians and nephrologists as well and come up with some sort of statement or guideline that you as a patient may be able to take to your nephrologist, especially a reluctant nephrologist, for them to read. So it might be helpful for us to do that. So David and I will work on that offline. I think something that you could put together and maybe
Definitely
run through some of our science advisory committee members.
Yeah, because there are growing databases on total kidney volume, and those are important for the research nephrologists to analyze, you know, the utility of total kidney volume and when in the course of disease. And the more data points that those databases have, the better for research in PKD.
Okay, so, and you may or may not know the answer to this, David, so it might be something that we have to follow up on, but someone is saying that on their MRI report, flow is reported. Do you have any idea what that is, what the implications are?
I know that I recently had an MRA, which is a magnetic resonance angiography, and that actually looks at blood flow. I'm not familiar with what flow might mean in an MRI.
So we'll follow up with that
Yes.
later. We'll look that up and let you know. Okay. So I think we've addressed, you know, I'll just reiterate, like David has said, you know, insurance may or may not cover the cost of these tests. I think it probably would depend on your particular insurance. So it's something that you would certainly want to look into before you necessarily demand these, or if it's not covered, you would want to make sure that you're able to cover it financially on your own.
And I would also like to add that these imaging techniques are often used for other reasons. Matter of fact, they're mostly used for other reasons. Your nephrologist, for example, if you have acute pain and you have PKD, they may want to look to see if there are signs of infection that can be detected by imaging, or if there are signs of bleeds into a cyst that can be detected by imaging. So imaging is often ordered for reasons having nothing to do necessarily with total kidney volume, although once those images are taken, total kidney volume could be subsequently calculated. But imaging is obviously used for a lot of reasons in renal disease, and it may not be just for the measurement of TKV.
So that's actually great, and we got a question via Twitter wondering if a person has an old MRI from checking, it was actually the MRI that diagnosed their PKD, can they use that image to determine TKV?
I am told that it can, that there are mathematical ways to gain information from that scan to estimate total kidney volume, so that yes, those are useful. I've actually been asked to see if I can get the data from my MRI from a while ago to participate in an observational study. So yes.
Very good. Okay, so if you have larger kidneys because of your height, would it mean that, let's see, if you have larger kidneys because of body height, does it necessarily mean your cysts would be larger than a person shorter in stature? So does your TKV, is it correlated to your body size, height, weight?
So I think there is, in general, a correlation between height and kidney size, even without PKD. I think height probably indicates that you will have more space for your growing kidneys, for one thing,
right,
and may not show that growth as obviously as somebody shorter in stature. And actually, kidney height is roughly correlated to total kidney volume. So if you want a really quick, simple, crude estimate of total kidney volume, you can extract that from the height of a kidney, although the standard deviation, as we talked about, will be much larger with those kind of measurements.
Great. Okay, so can you go back actually to the second time you used the Torres graphic with yeah, right there. Okay, so there's a question about that, and where is it? Okay, can you estimate the GFR in the yellow box and in the red? Is there a way to do that, or are we, you know, this is really more about TKV, not so much about its size, not function, so what, can you comment on that?
So just as you can measure total kidney volume at any point in time, you can measure estimated glomerular filtration rate at any point in time. And one of the easiest ways to do that is by serum creatinine, which is a simple blood measurement. Now, just like MRI, there are more sophisticated ways to measure GFR than serum creatinine, but that's a pretty good rough estimate of kidney function. So, and obviously a lot cheaper than MRIs. So pretty much in every blood test that you would take for a physical, for example, creatinine is measured, and you can get a rough estimate of kidney function.
Okay, so now go back to the one where you had the two pictures of the cystic kidney and the football and the cystic kidney and the typical kidney. Okay, so this one, I don't know that we can answer this beyond speculation, but someone was wondering, I believe, if the photos of the cystic kidneys were taken from, you know, where did they come from? Where did they originate? And I don't know. I would believe that probably it was part of our tissue donation program, and we work with a couple different institutions to receive that. So I would assume that these were taken from a nephrectomy. So they were removed from a patient via nephrectomy. And I don't know that we know that for sure.
Yes, and that's obviously in a nephrectomy, you know, a pathologist will look at the kidneys grossly. These are gross specimens, and then they will actually look at them histologically as well.
Oh, and actually, I guess they were asking about this also from the other photo with the variability photo. Mm-hmm. So again, we're not sure if they were from a live person via nephrectomy or from a not alive person.
Right. it's difficult to tell just from the photos.
Yeah. So is there you guys have really great questions, and we are not going to get to them all, but I will work with David to answer them. There are just some really good ones, and they're still coming in, and I love that. But oh, someone followed up the GFR. What's the GFR in the yellow box? Was the follow-up to that?
So very crudely, I think in general a GFR of greater than 60 is considered normal, but normal is variable. So large people may have a GFR of 120. That's their normal. Other people may have a GFR of 70. That's their normal. So it really, just like total kidney volume, there's a standard deviation in terms of what's normal for you as compared to somebody else. But I think in general, once it falls below 60, that it would be considered abnormal. So again, in the yellow box, you would be getting more variability than in the green box. And in the red box, you again have more variability as people's GFR is changing more rapidly over time.
Okay, so someone is asking, is TKV measurement standardized?
There are several proposals for standardization. Some of these are in Dr. Torres' lab, for example, at the Mayo. I think they have a totally automated system for taking MRI imagery and calculating TKV. There may be institutions that do it in their own way, but standardization is important, particularly for scientific studies. So I would imagine in clinical trials that are ongoing and in future clinical trials, the investigators will settle on something similar to what's being done at the Mayo, so that the same techniques and technology are used for all patients in the study.
And I will say that participating in a clinical trial, if that's something that you are interested in doing or you choose to do, is an option for getting, could be an option for getting MRI scans regularly and having your TKV measured. So there are some trials happening now and coming online very soon that may include that test as part of the study. So it's something to keep in mind, and you can find information about clinical studies and trials happening on the website at pkdcure.org.
And it's one of the reasons that trials are expensive, whether they're for PKD or other diseases. Multiple MRIs begins to add up, and the drug companies, of course, are paying for those, not the patient.
Okay, I am going to ask, I'm going to go 1 or 2 minutes over time because there are just 1 or 2 more questions that I really want to ask, and then we'll close down. And I know that not everyone has had their question answered, but we will try to answer those offline. So one person has asked a personal question of you, David, and I know that you won't mind sharing this. They want to know what your TKV is. So could you explain your situation?
Wow. I had my transplant and double nephrectomy done at Northwestern University in Chicago. I don't know what my total kidney volume was. My wife was told when I was still doped up after my double nephrectomy that they were like 2 boxes of swollen Wheaties cereal. But I never did get an absolute number, but I need to dig that out for the observational trial that I want to participate in.
Who would say that to you? Or your
the surgeon.
Of course the surgeon would say that. Okay, and then we've had someone, I just lost them. Well, okay, so completely sort of aside from TKV, more asking, is there something to do to help slow the growth of your TKV? There's really no guideline from a nutritional standpoint as any diet or anything you should limit that can have a scientifically proven effect on TKV. So nothing like caffeine or protein or anything like that, correct?
Well, I think in general, a renal diet, quote unquote, which stresses smaller amounts of higher quality protein, preferably protein from sources other than meat, are important. And the role of water has been a long-standing topic in PKD research. And there are studies ongoing that can actually calculate the amount of water you should drink given what your diet is. But that's research that's ongoing. But I think that that research will probably be pretty valuable because, as I've mentioned in previous webinars, as a toxicologist, one is taught that all substances are poisons. It's just a matter of dose. And even water, believe it or not, can be toxic. For example, people who run marathons and drink too much water dilute the salt in their blood and can get into fatal cardiac arrhythmias. So everything, has its pluses and minuses, and that's why we don't offer advice at these type of webinars, and is really for discussion with your nephrologist.
Exactly. And you can look for some information in the upcoming PKD cookbook. There will be a little bit of information in there about water, about diet, about caffeine intake, things like that, and that will be available in late June after convention. It's a brand new piece, and you can purchase it from our store. It's coming soon. Okay, so I'm gonna close it down there. Someone asked about the observational trial you're going to participate in, so we will put that information along with answers to the rest of these questions on the website in the next week or two, as well as in the next few days we will post the recording of this webinar. And thank you so much, David. This has been a really great webinar. I've learned a lot. I'm sure everyone else has too. There's been great engagement and good questions, so all around, really good one. And we are going to just, at that, say thank you everyone who participated. Don't forget to stay tuned next month where we will have a conversation with a PKD researcher. Look for information about that in your email. And also the PKD National Convention in June. It is getting very close. We're getting very excited here in the office. David will be there. I hope you will too and have a chance to meet him in person. Thanks a lot, everybody. Have a great day.
Bye-bye.